Hi, I'm Renee King, and the title of my project is an assessment of biologic. Sorry. Okay. Hello, my name is Renee King, and the title of my project is an assessment of biodegradability of 17 alpha ethylene estradiol and nanophenol, or EE2 and NP. EE2 and NP are trace organic contaminants, which are a subset of endocrine disrupting compounds. So endocrine disrupting compounds are agents that interfere with the synthesis and the binding of hormones in the body, which just means that these trace organic contaminants have the same effect on the body if they're intaken by humans or wildlife. So if these torcs are found in water after it's been through a wastewater treatment plant or a water reuse, then if traces are still found, it has a negative effect on the environment. I wanted to use the method of biodegradation to see if these chemicals can be broken down, because biodegradation is just the biological transformation of organic chemicals to another form. So to do this, I wanted to use 3 different forms of wastewater treatment plants. They were nitrification, denitrification, and anammox. The difference between these processes are shown in the graph. For nitrification, the ammonium oxidizing bacteria break down the ammonium sulfate to nitrite, and the nitrate oxidizing bacteria break down the nitrate to nitrate. From there, an extra organic matter source, such as methanol, needs to be added for it to be broken down to nitrogen gas so that it can be released back into the environment. For the denitrification process, nitrate is converted to nitrite using the same method with the addition of methanol. The difference between these 2 forms and anammox is that through the anammox process, ammonium oxidizing bacteria have the ability to take ammonium sulfate and nitrite and convert it straight to nitrogen gas without the extra organic matter. So these are just a picture of the chemostats that I used, or the reactors that I used. This is the anammox reactor where the bacteria was cultivated. This is the denitrification reactor where it was cultivated, and the nitrification reactor. Both the denitrification reactor and the nitrification reactor had to be started up. So to make sure that the reactors were functioning correctly, I had to do the performance test, or to test the performance of the reactor. I conducted nitrate, nitrite, ammonium, chemical oxygen demand, and specific denitrification rate tests. In these graphs here, it's shown that the nitrification chemostat was able to break down the ammonium that was input into the reactor to almost a 0 concentration for most of the period of time that I conducted my research. However, for the denitrification reactor, I came across a lot of problems. A lot of them were unknown, but with the bacteria, it didn't become stabilized until more towards the end of my research. Once both reactors were at a stable point, I was able to actually conduct the biodegradation test. Through these tests, I would take out samples from each of the reactors, and to create a control, I inserted NaN2, or sodium ASide. And this was used to stop the reaction of bacteria. So for the results of this test, the nitrification reactor broke down the trace organic contaminants at the most over the period of time that it was tested, which was over a period of 5 hours. For the NP concentrations, the denitrification reactor broke down the chemicals the best over the period of time. So from this, I was able to see that the nitrification and denitrification reactors did work the best. However, I noticed that the anammox reactor was actually able to break down the trace organic contaminants, which I did not foresee happening because it did not use extra organic matter source, and it was just bacteria in the reactor. So in the future, if I was able to actually optimize a way to have the bacteria break down the trace organic contaminants from the anammox bacteria, if I was able to find a way to optimize the anammox bacteria to break down the trace organic contaminants at a faster rate, I think this would be more productive in a larger scale wastewater treatment plant, because it takes away the cost of an extra organic matter source. Thank you.